Hello students, welcome to the biology class. So in the last class we finished the concept of the amino acids. We finished the concept of the protein, its structure, and how are the various levels of structure we understood. We even understood the various classifications of proteins possible according to the various criteria or basis. So now let's finish this concept with the last topic in proteins, and that is the functions of proteins. So there are various types of functions. I have jotted down few which are the easiest one or very easy to be understood. So the first one we can take up the concept is the sweet protein. We all know there are people who are diabetic in this world, and the doctors will start suggesting them stop or reduce the intake of your sugar or carbohydrates and all. So but the taste is something which we always we enjoy. When we consume tea, when we consume these things which are sweet in taste, we always crave for such type of things. So for such patients, the medical science has come up with the concept of sweet proteins. Now what are sweet proteins? There are some proteins which are sweet in their nature, and their sweetness is even more than sucrose. If you remember, we have learned this already. Sucrose sweetness index in the topic of monosaccharides and disaccharides, I have explained this concept, that the sweetness index is 100 in the case of sucrose or the table sugar or the cane sugar. But when we talk about some proteins, there are some proteins which are even sweeter, 200 times, 2000 times sweeter than the sucrose. And examples are monalin, brazein. If you remember these names, I think I took before also, that these are African plants or bushes. These are bushes from the root system especially, the sweet proteins are extracted and they are processed, and finally they are sold in market for those who are diabetic, because they are sweet, so they will at least give some taste to the food they consume. But remember, they are costly obviously, because extraction process is a very costly one. And then remember, we have already learned before, there are some sweet amino acids also, and we learned the concept of glycine and alanine. So in the same manner, there is a dipeptide also, but remember, it's not natural, it is artificial. That means in laboratory, scientists have created a dipeptide which is called as aspartame. Aspartame is a combination of only two amino acids, aspartic acid and phenylalanine. So when these two amino acids join together, this synthetic dipeptide is sweet, and it is 200 times sweeter than the sucrose. So these are the sweet proteins which help those who are diabetic. So their taste can remain in food without adding carbohydrates in their body, so their sugar level will remain normal. The second function we can learn is that the proteins can act like buffer. Now what is buffer? We have already learned in the last classes also regarding amino acids. Buffers are those things which are able to maintain the pH. So if it is alkaline or if it is acidic, it will remain as such. So proteins function as buffer against both the alkaline as well as the acidic chemicals, and therefore maintain a proper pH of the cell. That means a protoplasm of the cell maintains the pH with the help of proteins. So proteins present in the protoplasm act like a buffer also. Third property we can understand is the transport across the membrane. We all know permeation means ability to allow something to go across, maybe towards the inside or towards the outside or both directions. So membrane permeability or transport occurs because of protein. Now this brings us to the topic again of the fluid mosaic model. If you remember that model in the 8th chapter we have done, that there is a bilayer of there are two bilayers of phospholipid, and in between there are two types of proteins present, integral, and the second one was the peripheral. So peripheral proteins are those which are present either on the outer side or inside, extrinsic or intrinsic, but there are those proteins which are present throughout the width of the plasma membrane, and they are called either as the tunnel protein or carrier protein. If a transport protein is like a tunnel and allow things to go from that middle portion, there is a space or a cavity which is called as the channel protein also. So such proteins are called as one type of transport protein. Second is a carrier protein. Let me draw a small diagram to explain. Imagine this is the plasma membrane. So there will be a tunnel type of protein. This protein allow things to go across, but remember, size matters a lot. If the molecule trying to move is larger in size, this tunnel won't allow its transport. So it is size specific always in nature. In the same manner, carrier proteins are there. Now those proteins which are situated in such a manner that they cover the whole plasma membrane, but they have a habit of picking up, so they have a point from where entry of a compound is possible. Maybe a mineral ion is present, so it picks up a compound, takes 180-degree turn, and then release the compound from the opposite side. Such type of proteins are called also as transport protein, but which category? Carrier protein. So two types of proteins are possible which help, two types of proteinaceous structures are there for the passage of the solutes across the plasma membrane, and they are ion channels, or the other one can be called as the carrier proteins. So ion channels or tunnel proteins and the carrier proteins. The fourth property or fourth function possible is the iron stored in proteins. I have told in the last class also that it's a type of metalloprotein, that iron can be stored in protein, and such a protein is called as ferritin. So iron plays an important role in the animal body, and iron protein complex is called as ferritin. Another function possible is the osmotic potential or osmotic pressure to be maintained. We all know the amount of blood in our body is fixed. This fixed amount of blood may change because the maximum portion of the blood is the plasma, or you can say the maximum portion in the plasma is the water. So water amount continues to fluctuate in our body because sometimes we are sweating, sometimes we are drinking less water, or sometimes the amount may increase also. Then we will pass out more urine or more sweating. So how to maintain this osmotic potential or osmotic condition of our blood is by the help of some proteins, and those proteins are able to maintain osmotic conditions inside the blood plasma. And the best example for this is albumin. You'll be learning about this more in the circulatory chapter. So